L-name hydrochloride, also known as Nω-Nitro-L-arginine methyl ester hydrochloride, is a pharmacological compound that is commonly used in scientific research to study the role of nitric oxide, NO, in various physiological and pathological processes. It is a potent inhibitor of nitric oxide synthase, nNOS, the enzyme responsible for the production of nitric oxide. Mechanism of action. L-name hydrochloride works by inhibiting the activity of nNOS enzymes, specifically the endothelial, eNOS, and neuronal, nNOS isoforms. These enzymes convert the amino acid L-arginine into nitric oxide, a signaling molecule involved in numerous physiological functions, such as vasodilation, neurotransmission, and immune response. By inhibiting nNOS, L-name reduces the production of nitric oxide, leading to altered cellular signaling and physiological effects. L-name hydrochloride is commonly used in cardiovascular research to study the role of nitric oxide in blood pressure regulation, endothelial function, and vascular tone. By inhibiting nitric oxide production, L-name can induce hypertension in animal models, allowing researchers to study the effects of reduced nitric oxide levels on the cardiovascular system. L-name hydrochloride is also utilized in neuroscience research to investigate the role of nitric oxide in neuronal signaling, neurotransmission, and neurodegenerative diseases. By inhibiting nitric oxide production, L-name can modulate synaptic transmission and affect neuronal function. L-name hydrochloride is used to study the involvement of nitric oxide in the immune response and inflammatory processes. Inhibition of nitric oxide production by L-name can impact immune cell function, cytokine production, and inflammatory signaling pathways. L-name hydrochloride is typically administered via intraperitoneal injection or added to cell culture media for in vitro studies. The dosage and duration of treatment vary depending on the specific research objectives and experimental design. It is important to note that L-name hydrochloride should be used with caution and under the guidance of experienced researchers due to its potent and long-lasting effects on nitric oxide production. L-name hydrochloride can have systemic effects on various physiological processes due to its inhibition of nitric oxide production. It is crucial to carefully monitor the dosage and duration of treatment to minimize potential toxic effects. While L-name is a widely used nNOS inhibitor, it is important to note that it may not be entirely selective for nNOS enzymes. It can also inhibit other enzymes, such as arginase, which can affect L-arginine metabolism and potentially confound experimental results. The response to L-name hydrochloride may vary among different species, and caution should be exercised when extrapolating results from animal studies to humans. In conclusion, L-name hydrochloride is a pharmacological compound used in scientific research to inhibit nitric oxide synthase and reduce nitric oxide production. It is commonly employed to investigate the role of nitric oxide in cardiovascular function, neurological processes, and immune response. However, its potent effects and potential non-specificity require careful consideration and appropriate experimental design.